Hi, I'm Tess. I'm a second year master's student in Dr. Roberts' lab. I'm going to talk to you about moss development on Tuesday. We'll be working in this room, which is our sterile culture room. Here is our sterile hood. It's where we culture moss so that it isn't contaminated with bacteria or fungi. We're going to image that moss on that microscope, and we're going to look at moss through various stages of development. So on Tuesday, I want you guys to be able to tell me what's the difference between different moss developmental stages, between protonema and gametophores, and between protonema and protoplasts.
Hi everyone, I'm Professor Norris, and I'm going to set up a station for your field trip where you're going to see gel electrophoresis of DNA molecules. And so this is an area where we run gels in these gel apparatuses. So you're going to get to see a gel, actually feel what a gel feels like, and then we'll practice loading a gel because you have to load the samples in these tiny little wells. So you get to try doing that yourself. I'll also show you some data from gels that have been run so that you can see what the DNA bands look like on the gels. This big box near me here contains a camera and a special apparatus that allows us to see the DNA bands with a stain called lithium bromide. So I have 3 questions I would like you to think about and try to answer before you come for the field trip. The most important question is why do DNA molecules move on this gel? Why do they travel from the top of the gel to the bottom? I want to know why did they move when we turn on the current. I think you can see these cords here. This is connected to this apparatus. This gel is actually running, I just realized. And so, when we turn on the current, the molecules move from the top of the gel to the bottom. So the first question is, why are they moving? Second question, the gel is made of something called agarose, agarose. I want you to find out where agarose comes from. And the third question, when I have you load your samples, the sample will sink to the bottom of the little space, the little well, and not mix with the water. So I'd like you to find out why the sample sinks and doesn't get diluted into all of the buffer and volume of the gel. So why does the sample sink? And that's it. I'll see you next week.
Hello everyone, I'm Dr. Roberts. Welcome to our microscopy room. When you come for your field trip, we'll be telling you about the different kinds of microscopes we use. The one that you see over here on my left is called a stereo microscope. And there's another one behind me, I think you can see it over here. That's called a compound microscope. And before you come for your field trip next week, I'd like you to find out what the difference is between a stereo microscope and a compound microscope. We look forward to seeing you next week.
Hi, my name is Mai Tran. At this station, me and my partner Xing Xing, which you will see later, are going to talk about using a method to determine the amount of protein in each sample. And the method we're using is a BCA method. It's a protein assay kit where we can run a standard curve to measure amounts of our known protein sample, and then using that to calibrate how much proteins we have in our unknown samples. And a question that we would like to ask you at the end of the demonstration is, how does this method work, and what's the mechanism behind the color change that happens during our protein determination method?